Welcome to The Explainer. Today, we're going to take a journey deep inside our own cells to answer a really fundamental question. How does the food we eat actually become the fuel that keeps us alive?
I mean, think about it. How does a single meal power the trillions of cells that make you, well, you? From the neurons firing in your brain to the constant, steady beat of your heart, it all comes down to one tiny, universal molecule. It's the energy currency for pretty much all known life on Earth.
Meet ATP, or adenosine triphosphate. The easiest way to think about it is like the cell's own rechargeable battery. And this is the key takeaway. Our bodies don't just, burn food for fuel. Instead, they perform this incredibly controlled demolition of food molecules. And they use the energy that's released to charge up countless little ATP batteries. Those batteries are what power everything else.
Okay, so our journey kicks off in the main compartment of the cell, which is called the cytosol. A single molecule of glucose, a simple sugar, is about to pay its first toll on the energy highway, and this initial process is called glycolysis.
So glycolysis is basically the first splitting of that 6-carbon glucose molecule. What's really remarkable is that this process happens entirely without oxygen. That tells scientists that it's probably one of the most ancient metabolic pathways out there. It likely evolved on an early Earth way before the atmosphere was rich in oxygen. So it takes one glucose, splits it into 2 smaller 3-carbon molecules called pyruvate, and extracts a small but really quick burst of energy.
But here's the interesting part. The cell actually has to spend a little energy to make energy. In the first phase, the investment phase, it uses 2 of those ATP batteries to get the glucose ready. Only then, in the payoff phase, does it get its return on investment. 4 ATP molecules, plus 2 high-energy electron carriers called NADH. So that leaves us with a net profit of 2 ATP and 2 NADH. Now that small gain, that's just the appetizer. The 2 pyruvate molecules we just made are now heading off on a journey into the true powerhouse of the cell, the mitochondrion. This is where the real, heavy-duty energy extraction is about to begin.
Before it can enter the next stage, each pyruvate molecule gets a little bit of a makeover. A carbon atom is stripped off and released as carbon dioxide, and yep, that's part of the very same CO2 you're exhaling right this second. The 2-carbon piece that's left over gets attached to a carrier, and this forms acetyl-CoA. You can think of this molecule as the key that unlocks the next big phase.
And that brings us to the Krebs cycle. It's like a metabolic wheel that spins twice for every single glucose molecule we started with. As acetyl-CoA enters, its carbon atoms are systematically broken off and released as more CO2. But the real goal here isn't to make a bunch of ATP directly. Nope, the main point is to harvest high-energy electrons, loading them onto our carrier molecules, NADH, and a similar one called FADH2. So let's just hit pause for a second and see where we are. After glycolysis and the Krebs cycle, we've completely broken down our original glucose molecule. But we've only made a tiny handful of ATP directly. The vast majority of the energy from that glucose is now being stored temporarily in these loaded-up electron carriers, NADH and FADH2. The cell has collected all this potential, but it hasn't actually cashed it in yet.
All right, now for the grand finale. This all takes place on the winding inner membrane of the mitochondrion. This is where all those loaded carriers, NADH and FADH2, are finally going to cash in their energy for a massive payout of ATP. The whole process is called oxidative phosphorylation.
So here's what happens. NADH and FADH2 drop off their high-energy electrons to a series of proteins called the electron transport chain. Evidence suggests that as these electrons get passed down the chain, their energy is used to actively pump protons across that inner membrane. This action creates this incredibly powerful gradient, a kind of stored potential energy that scientists call the proton motive force. It's a lot like building up a huge reservoir of water behind a dam.
Okay, so you've got this massive proton gradient, all this stored energy. But how in the world does the cell turn that into usable ATP? For a long time, the exact mechanism was a big puzzle. But the evidence now points to a process that's less like a simple chemical reaction and more like a stunningly elegant piece of molecular machinery.
It turns out, the cell uses this incredible molecular machine called ATP synthase. It's this complex protein that basically functions like a microscopic water wheel, or maybe a turbine. Its entire job is to convert the potential energy of that proton flow into the chemical energy of ATP. And this is where the process gets beautifully mechanical.
So current model suggests that as those protons rush back through a special channel in ATP synthase, they cause a rotor-like part of the protein to just spin at incredible speeds. This rotation creates a physical mechanical force which causes other parts of the enzyme to actually change their shape. And it's this physical change that's believed to literally squeeze ADP and a phosphate group together, forging a new ATP molecule. It is a true biological motor, cranking out the energy that powers our lives.
So what's the final count? What's the total payout? Well, biochemistry textbooks will often cite a theoretical maximum of about 38 ATP from one molecule of glucose. But the reality in living cells is a little messier. Some energy is always lost as heat, so a more realistic estimate is probably somewhere in the range of 30 to 38 ATP. Still, that is a colossal energy return compared to the too little ATP we got from glycolysis alone. So this entire incredible process relies on two key things: the glucose from our food and the oxygen we breathe. But that brings up an even bigger question. This can't be a one-way street, right? Where did that glucose and oxygen even come from in the first place? To find the answer, we have to look outside our own bodies and toward the sun.
The answer, of course, is photosynthesis. In plants, algae, and some bacteria, little organelles called chloroplasts carry out this process. And in so many ways, it's the beautiful mirror image of what we just saw in our own cells. This is where the cycle of energy for most of life on Earth really begins.
Photosynthesis happens in two main stages. First, in the light phase, chlorophyll captures sunlight to literally split water molecules apart. This moment right here, this is what releases all the oxygen we breathe into the atmosphere. This phase also generates some ATP. Then, in the dark phase, also known as the Calvin cycle, the plant cell uses that energy to capture CO2 from the air and build it into glucose, the very foundation of pretty much every food web.
The enzyme that makes this all possible is called rubisco. Its one job is to grab CO2 from the air, which is the first critical step in building a sugar molecule. This makes it arguably the single most important enzyme on the planet, and it might even be the most abundant protein on Earth, period. So the next time you eat something, just think about that complete journey. The energy in your food began as a photon of light from the sun 93 million miles away, captured by this one enzyme. It really reminds you that every breath we take and every meal we eat connects us to this vast planetary cycle of energy.